Welcome back, fellow mitochondriacs, for another episode of Cancer as a Mitochondrial Metabolic Disease. I want to continue our discussion talking about the importance of the deuterium hydrogen or deuterium proteum ratio when it comes to treating cancers and metabolic disease, and how deuterium depletion therapy works, how deuterium depletion therapy is achieved, and how deuterium depletion therapy can enhance or dovetail into metabolic therapy as a whole, or ketogenic metabolic therapy as described by Dr. Thomas Seyfried and his groups. So, without further ado, let's get into it. So, we left off our last discussion talking about the importance of deuterium depleted water, or probably more importantly, having our whole body deuterium level be at a certain concentration to achieve metabolic health. And I started the conversation out talking about its effects on this GLUT4 transporter and improving insulin resistance and ultimately lowering blood glucose, A1C, and hopefully your glucose ketone index, given that that is the true foundation of ketogenic metabolic therapy, is getting your glucose down, which is kind of how it dovetails into this larger series about lowering blood glucose to give you the tools so that you can get into ketogenic metabolic therapy and add the other therapies that make it such a kind of a beautiful, harmonious treatment protocol. So we talked about deuterium depletion therapy's effects on blood glucose. Now I want to talk about its effects on cancer as a whole, what the proposed mechanisms are. So the next paper I want to talk about is fairly recent, July of 2024, and it says the biological impact of deuterium and therapeutic potential of deuterium depleted water. And it says here, as an important compound of deuterium depleted water, DDW, possesses various special effects, including but not limited to altering cellular metabolism and potentially inhibiting the growth of cancer cells, demonstrating anxiolytic-like behavior. That would be like anxiety relieving behavior, enhancing long-term memory in rats, reducing free radical oxidation. Just remember that. It reduces free radical oxidation, regulates lipid metabolism, harmonizing indices related to diabetes and metabolic syndrome, and alleviating toxic effects caused by cadmium, manganese, and other harmful substances, implying its tremendous potential in anti-cancer, neuroprotective, anti-aging, antioxidant, obesity alleviation, diabetes and metabolic syndrome treatment, anti-inflammatory, and detoxification, therefore drawing extensive attention from research. And you see here that on this graphic, we see there that there's a distribution of deuterium in nature, and we've kind of talked a little bit about that, how generally there's less deuterium at the poles and there's more deuterium kind of at the equatorial level, and how also, and we'll go over it again, but also how, more carbohydrate-based foods are going to have more deuterium, and the higher fat and protein foods are going to have less deuterium. And it's talking about at the molecular level, looking at DNA, the organelles, and such as mitochondria, cancer level, et cetera, et cetera. And then it's showing like the role of deuterium depleted water and certain diseases, whether it be an anti-cancer effect, whether it be a neuroprotective effect, an anti-aging, an antioxidant effect, which again, I think I want to reiterate, an antioxidant effect, others. So why do I bring that up? The antioxidant effect? Well, because as we talked about multiple times in the past, generally, we don't want to be giving antioxidants for cancer because that potentially could hamper or even damage metabolic therapy. But sometimes, and it's above my ability to explain why, but sometimes these therapies can have an antioxidant effect within normal cells or maybe not as damaged cells, and then within cancer cells, particularly, they have an opposite effect, which we will see. So I just want to highlight that. We've seen that happen with many other compounds: resveratrol, curcumin, sulforaphane, a lot of other compounds, melatonin, etc., vitamin D. They all have an antioxidant effect in kind of normal or pseudo-normal cells that are non-cancerous, non-transformed cancer cells. But then when it's a cancer cell, it has an opposite effect. It actually inhibits our antioxidant response. It creates free radicals. So it's just very fascinating that deuterium depleted water, as we'll see later when we go through this, has a very similar effect. So I pulled this graphic only to show that this is where this information that I'm going to be talking about next comes from. We showed this graphic on the very first part of talking about deuterium. So I wanted to talk about this. So this is part of the paper that I clipped out here, and it says the effect of DH shift on proliferation and apoptosis in tumor cells. So that's the deuterium hydrogen shift. And it says, in addition to activating effects of isotopically altered water described in various studies on healthy cells and body tissues, a number of scientific papers have investigated the effect of DDW and DEW, deuterium-enriched water, in neoplastic or cancer and metabolic disorders. A large proportion of in vitro and in vivo and clinical studies have demonstrated the anti-tumor effects of deuterium depletion of cells. A recent study showed that reducing concentrations of deuterium in the cellular environment to 30 to 100 parts per million can lead to regression of a breast cancer or breast mammary tumor. Reduced deuterium water has been shown to affect the cell cycle, decrease the number of cells in the S phase, and significantly increasing the populations in the G1 phase, as well as inducing antioxidant enzymes. A similar effect was previously demonstrated on nasopharyngeal carcinoma cells in vitro and determined with a deuterium concentration of 50 to 100 parts per million. DDW significantly inhibited proliferation and reduced colony-forming invasion capacities of the cells. There was a cell cycle arrest with a decrease in number of the cells in the S phase and a significant increase in number in the G1 phase. It is particularly interesting that instead of growth inhibition, DDW promoted the growth of normal control cells in the same experiment. So this is actually fascinating, and I think I may have missed this last part when going through this paper initially. So not only does it have an antioxidant effect within normal cells and a pro-oxidant effect, which we'll talk about later, within cancer cells, it actually has a growth inhibition effect on cancer cells, and even a growth kind of it promoted growth within normal cells within the same experiment, which I thought was interesting. Now, we talked a lot about phases, and basically this graphic just kind of going over this. So there's the actual mitosis, or the separation of the two daughter cells, right? But then there's these growth phases, right? So what they're saying is that when you give deuterium depleted water, it kind of stops this S phase, this deuterium, this DNA synthesis phase, which is needed to create new daughter cells. It kind of arrests the cells in an area where it's not able to grow within cancer cells if the deuterium concentration is lower within those experimental models. It later says that thus the reduction in deuterium concentration in the large environment was able to regulate the cell cycle of both normal and tumor cells. It is known that the transport of hydrogen ions, I'm going to skip this, let's get on a little bit further. Later investigations have shown that the incubation of malignant cells in deuterium depleted water for 48 hours results in this is a key, results in a disbalance between reactive oxygen species production and neutralization in mitochondria, thus leading to oxidative stress in the cells, which activates apoptosis, but the effect was pronounced in some, but not all, studied malignant lines. Later says that a possible mechanism of deuterium depleted water interference in the cell cycle may also underlie the downregulation of expression of certain genes. Some researchers attribute the suppression of tumor cell proliferation to the direct effect of deuterium depleted water on the level of activation of pro-apoptotic and anti-apoptotic proteins, For example, several studies have clearly demonstrated that decrease in gene expression of BCL2, KRAS, and CMYC proteins in various tumor cells. So we're going to talk about KRAS and CMYC in particular in the coming slides. We've touched on it throughout this series, but I want to reiterate why those particular proteins are important at downregulating when it comes to the kind of cancers and metabolic disease process. The effects of reduced deuterium levels on the organism on mitochondrial function have also been shown in some studies. The transformation of cells into malignant cells is also associated with the development of mitochondrial defects. We know that mitochondrial heteroplasmy that results in a lack of hydration of the tricarboxylic acid cycle intermediates with low deuterium matrix water. All oncometabolites not related to the TCA cycle arise from aerobic glycolysis, glutaminolysis, or single carbon metabolic cycles. So what it's saying here is that what it's saying here without saying it is that one of the, I don't want to say mysteries, but one of the curious things about mitochondrial energy production and using the TCA cycle is that there's a lot of steps to do not very many things, or to transform a molecule to a not very much, right? And what they hypothesize is the reason why there's so many steps in the TCA cycle is not necessarily the biotransformation of the molecule, because we need that many enzymes to transfer, go to pyruvate to the end of the TCA cycle, but to gradually remove deuterium from the mitochondrial matrix and then allow the maximum amount of protium or light hydrogen to get into the electron transport chain so it doesn't mess up the electron transport chain and the nanomotor, the ATPase. So what they're saying here is that because in cancer, all oncometabolites not related to the TCA cycle arise from aerobic glycolysis or the Warburg effect or the breakdown of glutamine, glutaminolysis. Again, this is a completely other area of science. Dr. Gabor Sommelier and his groups and all these other groups, this is not coming from Seyfried's group or Seyfried's lab, right? These are like completely different areas of science. And they all are telling us that the Warburg effect and glutaminolysis, or the breakdown of glutamine, is where the majority of the energy is made within a cancer cell, right? So just another reason why, like, I stick towards talking about the Warburg effect and glutaminolysis, not because I necessarily have hero worship of Dr. Seyfried, although I do think he's amazing. It's because that's where the majority of other areas of literature are confirming that this is how cancer metabolizes, certain substrates, glucose and glutamine. I'll digress. Deuterium depletion during metabolic water production is one of the most important functions of the mitochondrial matrix. It has been hypothesized that in tumor cells, the balance between the activated H+ transport system and DDW, deuterium-depleted water, producing mitochondria, which determine the DH ratio in the cells, is disturbed. This disruption leads to an accumulation of deuterium in the cells and basically causes the genetic cancer defects that we see. And it also changes the size and function of nuclear DNA. Reducing the concentration of deuterium in the cell due to the consumption of DDW restores the disturbed DH balance and deeply changes the cellular phenotype and proliferation, which I think is very fascinating. I know we mostly talk about, cancer as a mitochondrial metabolic disease, and we've talked about how, how do I say this? We've talked about how TCA intermediates can likely, change gene expression as a way for the mitochondria to control epigenetic phenomenon or to change the gene expression within the nucleus. We've also talked about retrograde signaling and some of the mitochondrial portions of our series, and it's about how they use different reactive oxygen or reactive nitrogen species as a way to signal to the cell that we need to upregulate certain genes or antioxidant response elements or so on and so forth as a way to maintain homeostasis, redox homeostasis within the cell. But this is like another level of, I guess you'd call it retroactive signaling, because when there's damage to the mitochondria and the TCA cycle is not able to get the deuterium out of the cell, and then the deuterium breaks the nanomotors and increases reactive oxygen species, and that controls the entire cell's deuterium hydrogen ratio. That will then, in addition to further breaking mitochondria and leading to reactive oxygen species and oxidative stress and further mitochondrial damage as a kind of a vicious snowball, it will also, that DH ratio will also, very importantly, change gene expression to lead to a more cancer type phenotype of a cell, if that makes sense. So it will genetically reprogram the nucleus to be more like a cancer, cell. So it's like another level of how mitochondria, when they're broken, they can change gene expression and lead to cancer, if that makes sense. Several in vivo studies have also confirmed the anti-cancer effect of lowering deuterium levels in the internal environment. For example, a double-blind randomized 4-month clinical trial, stage 2 of different locations, the administration of DDW caused a 7-fold increase in the median survival time of lung cancer and a 2-fold increase in breast cancer, and effectively prevented early stage breast cancer recurrence. In addition, previous clinical studies on patients with various types of tumors showed consumption of 10 to 20 parts per million DDW caused the arrest of malignant cell growth and significantly increased patient survival, as well as improving quality of life. In this regard, some authors suggest that water with reduced deuterium concentration can selectively affect tumor cells due to its ability to regulate the key genes in the cell cycle without, however, interfering with metabolic processes in normal tissue, which is kind of the goal, right? We want a therapy that's going to effectively improve normal cells, make them healthier, and then selectively target cancer cells, right? And that's why I'm so interested in phytochemicals and things like methylene blue and red light therapy and, vitamin D and melatonin, because it seems that they have a selective understanding of what requires, killing off and what requires kind of reinforcing or boosting. And again, DDW, or deuterium depleted water, seems to be one of those other interventions that is very similar. So kind of going into the mechanism, I've already alluded to it when we talked about cell cycle and gene expression and retrograde signaling to the nucleus, et cetera. And we've kind of touched on the idea that the DH ratio can lead to redox instability or redox imbalance, and lead to oxidative stress. But it hasn't been very clear, because I've also talked about having an antioxidant effect. So I want to make it very clear that it does not have an antioxidant effect within cancer cells. It has actually an opposite effect, and that's what I show with these next couple of papers. So despite the convincing empirical evidence that deuterium depleted water, DDW, somewhere between 25 and 125 parts per million, has anti-cancer effect, the molecular mechanisms remains unclear. Here, redox proteomics investigation of the DW action and this A547 cells revealed an increased level of oxidative stress, whereas expression proteomics in combination with thermal profiling uncovered crucial role of mitochondrial proteins. In the proposed scenario, reversal of the normally positive deuterium gradient across the inner mitochondrial membrane leads to an increased export of protons from the matrix to the inner membrane space and an increase in the mitochondrial membrane potential, enhancing production of reactive oxygen species. The resulting oxidative stress leads to a slower growth and can induce apoptosis. This gets me back to a very important concept that we've kind of had to hit over and over again that is somewhat difficult to understand, I think, for everyone. And that is, why would we want to potentially enhance mitochondria within cancer cells. The mitochondria are broken. They're definitely broken. They're vacuolated. They have, they're not able to produce energy through electron transport to maintain any kind of cellular function. They only are there as kind of empty vessels. And what Dr. Seyfried calls cancer mitochondria is he calls them ghost mitochondria, because when you look at them under an electron micrograph, all of what makes the structure and function of a mitochondria effective and efficient and work is absent in a mitochondria and a cancer cell. So they're not doing much. And as a matter of fact, if you look at the biology of, and I have several videos about this, but and it's talked about through the entire series when it comes to like dichloroacetate, thymine. I talked during melatonin as well. But under the influence of hypoxia-inducible factors, HIF1 and 2, basically energy flux or energy going through the mitochondria is stopped at the level of pyruvate dehydrogenase complex, PDC, or pyruvate dehydrogenase, through a protein called PDK, pyruvate dehydrogenase kinase. And that effectively shuts the energy down from going through a mitochondria that's broken. And one of the reasons why I believe that happens is because it doesn't want energy to go through the mitochondria, because it's going to create massive amounts of reactive oxygen species. Well, why would cancer want to prevent that? Well, because that's how, in general, at least one mechanism of how mitochondria set off apoptosis or programmed cell death. And cancer cells obviously want to evade programmed cell death. They don't want to die. They want to remain immortal. And that's what the Warburg metabolism allows them to do. They get out of mitochondrial's control of programmed cell death. So one of the reasons why deuterium depleted water works is because it, as it says here, is it increases reactive oxygen species on the inner mitochondrial membrane, and it restores apoptosis or programmed cell death within cancer cells, which has been shut down. One of the ways we've seen this being done in the past by talking about these mechanisms is through things like PDK inhibitors, or pyruvate dehydrogenase inhibitors, or even shutting down hypoxia-inducible factors is kind of a back-end way of doing it as well. But what we're doing is we're restoring flux through the pyruvate dehydrogenase enzyme. And I don't really have a picture here, I don't believe, unfortunately, like close at hand, or I'd show it to you. But melatonin has an effect on this. Vitamin D has an effect on this. But the prototypic inhibitors of PDK is dichloroacetate. And then I've talked about how thymine has a very similar effect when it's in high doses as dichloroacetate or DCA. That's one way to increase reactive oxygen species within cancer cells, cancer mitochondria, to restore programmed cell death. The other way was looking at red light therapy. Red light therapy also will improve reactive oxygen species production within a broken mitochondria to restore apoptosis. That's one of the proposed mechanisms how it works. And then methylene blue and deuterium depleted water also have a similar effect, right? Where they're basically increasing reactive oxygen species within the mitochondrial electron transport chain, which ultimately unlocks apoptosis. So that's kind of in line with what we've already talked about from several different therapies in the past. But deuterium depleted water is a very interesting, different way of doing it. So I'll digress. So again, what is actually happening, based off of what that paper is actually saying, right? So I talked about it a lot, and reading the abstract, but when we actually go and look at this graphic, it kind of helps us understand the mechanism a little bit further. So if we are drinking, normal deuterium water, or if we have broken mitochondria and the deuterium concentration is higher than normal because the regulator is a functioning mitochondria, and the deuterium is actually aggregating or concentrating within these cancer cells, then that will help lower the amount of reactive oxygen species within these broken mitochondria. But when you go down to 80 parts per million, you see that there's an increase in reactive oxygen species. This is a superoxide reactive oxygen species type, and that will lead to the improvements in apoptosis and lead to cancer cell death. That's the proposed mechanism of how it works. However, it's interesting because what they found was is if you go too low on the DDW, if you go too low on the deuterium depleted water, you can actually have other mechanisms of protecting against reactive oxygen species. In particular, they were talking about this uncoupling protein that gets integrated into the mitochondrial membrane and allows for the kind of redox homeostasis, and actually kind of has a paradoxical not improvement effect. So that's why, if you look at Gabor Samoyed's book, they don't necessarily recommend starting with like 10 parts per million. I don't think I ever saw a podcast where he talks about it, or even anything that I've seen where they're like, start at the lowest possible deuterium concentration. Now, because what they're saying ultimately is that the ideal deuterium concentration for a human is somewhere around 125 parts per million. The reason why you would want to be more aggressive potentially, from what I can understand, is that the more aggressive you are on the front side, the more likely you are to lower your total deuterium concentration faster within a shorter period of time. Not necessarily that you want your total body concentration of deuterium at 10 parts per million. So if you were to start at 125 and be on a ketogenic diet, you're going to achieve 125 parts per million, but it may take a lot longer to get to that point, if that makes sense. So it makes more sense to be a little more aggressive than 125, or even like low deuterium naturally occurring waters, like like Evian or Voss or those, because you're not going to, it's not going to be enough to move the needle fast enough to get you to the outcome you're looking for. So I don't like giving recommendations, as you guys know, on YouTube, but I think a simple way to look at this would be to take, basically a 1-to-1 ratio of, the lowest deuterium, local deuterium depleted water, or whatever you want to call it. And then you would mix it like a 1-to-1 ratio with like a 10 ppm, and you'd get somewhere between like 70 and 80. And that seems to be a sweet spot, the 70 and 80, not only from the glucose side of things, but also from the kind of the sweet spot with the reactive oxygen species and activating apoptosis for these cancer models. So this is just like another graph showing that an increase in reactive oxygen species. And it was interesting because in this particular study, any reason, because it says here, in addition, we attempted to counter the DDW toxic effect by stimulating or simultaneous addition of NAC, or N-acetylcysteine, a thiol-reducing antioxidant agent. Both DDW and this other compound treatments, the suppressed cell proliferation was largely restored. So basically, any kind of anti-cancer, anti-proliferative effect of these two treatments were restored when you gave NAC. But also, as it says here, it says, gosh darn it, I think that later in the paper, it actually talked about how have their place in human health, and there has to be a redox homeostasis and a redox, because traditional metabolic therapy is trying to shut down glucose and glutamine as a way to shut down energy production, yes, but also to kind of tilt redox homeostasis in our favor, so that when we hit it with an oxidative therapy, it has no protection. And so what we'd be doing, same with deuterium depleted therapy, if we give antioxidants, we're going to basically, we're going to be literally drinking water that is pretty darn expensive, if you've actually ever, or in combination with chemotherapy, effectively inhibited cancer progression. In most experiments, the combination of treatment enhances the therapeutic effect of chemotherapeutic monotherapy. The inhibitory role of DDW in tumors is thought it is through regulating the reactive oxygen species, or ROS related genes, such as KEAP1. Further control normal amounts of ROS can inhibit the tumor progression. This particular paper, it was talking about like having a neuroprotective effect, or an antioxidant effect, or an anti-aging effect, is because it has antioxidant effects, and helps with reactive oxygen species, but within cancer cells, again, it has a. So why don't we get down into some nitty gritty, some clinical data?